dear students, welcome to another tutorial from Brilliant Bios. This is the third part of my vesicular trafficking lecture. In my first part, I have mentioned that vesicular trafficking is nothing but a secretory pathway of a protein, in which a protein enters inside the endoplasmic reticulum, then it travels through different Golgi apartments and then ultimately destined to the endosome or lysosome, plasma membrane or cell exterior. So in the second part, I have mentioned that this total process, this vesicular trafficking actually occurs by two ways. One is called vesicle budding, another is vesicle fusions. Only these two processes is responsible for the whole vesicular trafficking. As the name implies, vesicular, that means with the help of vesicles. And vesicles actually perform its functions by two ways. One is budding. It buds out from one compartment and then fuse with the other compartment. I will give you an example. Let's suppose a protein when bud out from the ear, then it has to fuse with the cis-Golgi network. The same is true when a protein bud out from the cis-Golgi network, it must fuse with the cis-cisterna. And the same process occurs throughout this whole process. So if you have understand this vesicle budding and vesicle fusion, you have understood the vesicular trafficking. In the second part of my lecture, I have discussed about vesicle budding, how vesicle budding occurs, what are the proteins are responsible for vesicle budding. Now in this class, I am going to talk about vesicle fusions. So I will give you some example of proteins that are responsible for vesicle fusions. The first protein you have to remember are called RAB-GTP protein, RAB-GTP. The second protein that is useful for this fusion is called V-snare. V-snare. And the third protein that is important for this fusion are called T-snare in capital. Okay. Now V for vesicle snare and T for tethering snare. I will draw a picture, then you will understand very easily. At first, you take the example. RAB-GTP is a protein that helps in the tethering of the vesicle to a particular site. There are different type of RAB proteins are present. Okay, so RAB1, RAB2, RAB9. Okay, so these are example of RAB proteins. I will draw a picture and I will show you how RAB proteins help in the fusions. Then there is some examples of V-snare, V for vesicle snare. So some proteins are present in the vesicles that help in the fusions. Example like VAMP, VAMP. Another example that is present in the neuronal synapse or synaptic vesicles. Example is synaptobrevin. Okay. Another example that is helps in this process also present in the vesicles called synaptotacmin. I will show you all these molecules. Then the example of T snares, tethering snares, examples are SNAP25 and syntaxin. Okay, so these are examples of T snares and these are examples of V snares. So now let me draw a vesicle and how this vesicle fuse, you can understand. Let's suppose a vesicle bud out from the ear and will fuse to the cis-Golgi network. So let me draw a cis-Golgi network. Let's suppose this is a cis-Golgi network. Okay, so this is a cis-Golgi network. So this is CGN. Now from the endoplasmic reticulum, let's suppose this is the endoplasmic reticulum. Vesicles are bud out. Okay, so budding has occurred. So these are the vesicles. This vesicle will fuse. Okay, so let me draw a big vesicles so you can understand. So let's suppose these are vesicles that will fuse with the membrane of the CGN, and these vesicles have some V snare like VAMP, VAM, but in case of synaptic vesicles, those carry neurotransmitter, they have synaptotagmin, synaptobrevin. So let's suppose this is synaptotagmin, this is also synaptotagmin, sorry, synaptobrevin. So in case of synaptic vesicles, those carry neurotransmitter, we call synaptobrevin, and other cases it is called VAMP, VAMP. And there is a helper. I have mentioned that there is a helper called synaptotagmin, especially in case of neurotransmitter vesicles, synaptic vesicles, but not present in this normal vesicular trafficking. So they look like this. They look like this. So this is synaptotagmin. So these are two synaptotagmin. Okay. Now this is called V snare because these proteins are present in the vesicles. So it is called vesicle snare, V snare. And I have mentioned that there is a wrap protein is present. So let me draw a wrap protein. Let's suppose this is a wrap protein. This is a wrap protein that is attached with the vesicles. So this is wrap with GTP. Now in the destinations where the vesicle will fuse, example is CGN, any compartment can it be. So let's suppose this is CGN, then the CGN will have a protein that will actually accept or receive this wrap GTP. So wrap protein helps in the tethering. So let's suppose there is a protein that is present in the CGN. We call this protein that actually attached with this wrap protein called wrap effector. Wrap effector. So wrap protein binds with the wrap effector and helps in the docking of the vesicles. Now these vesicles have V snare, it could be VAMP or synaptobrevin, and a helper hand called synaptotagmin in case of neurotransmitter synaptic vesicles, those carrying neurotransmitter. Now in the CGN or any other compartment, there are some T snares also present. Example, SNAP25 syntaxins, they are look like this. Okay, syntaxins and SNAP25, the three number, they are present in three number, SNAP25 2 and syntaxin 1. Now when they come close to each other, wrap effector proteins actually helps in the tethering or docking of the vesicles. They fuse, these all molecules actually fuse with each other. Okay, so there are three T snare and one V snare actually fuse, so they form a coil structure. These four molecules form a coil structure so that this gap is reduced. But in case of neurotransmitter or synaptic vesicles, unless or until calcium binds, calcium, this synaptotagmin is sensor of calcium, calcium binds with the synaptotagmin Ca+2, unless or until calcium binds, it will not fuse and will not release the neurotransmitter. Okay, but this is not case for this vesicular trafficking, but as this is the same mechanism of vesicular fusions of synaptic vesicles, I am mentioning here synaptotagmin. Synaptotagmin helps in the ultimate fusion with the membrane of the synapse. So forget about this. Only these molecules like V snare and T snare actually form a coil structure and brings these two membrane very close to each other that it will fuse with each other. And okay, so these are the very simple mechanism, but I have mentioned in my previous class and I also told my students that cell always try to reuse everything, reuse. So that's why if this V snare T snare form a coil structure, it cannot function another time. So for that reason, it should be at the same conditions as it was. So this coil structure should be actually removed. So for that reason, there is a protein responsible called that is called alpha SNAP, that is associated with a main protein called NSF. So NSF is the main protein with association of alpha SNAP. They two actually helps in this uncoiling, and ultimately what happens, this T snare will become single and the V snare also unfused. Okay, so this is the vesicle fusion. This is very important for your vesicular trafficking. You must remember the example, and these are two mechanisms. In this class I have discussed about vesicle fusion. These are two mechanisms, and the same mechanism occur repeatedly. Ultimately the protein reach to the destination endosome, lysosome, plasma or cell extrusion. So thank you for watching, and always keep learning.